Welcome to video 1 of my final project. In this project, I kind of want to talk about depolarization, muscle contraction, how we're actually exerting force in the heart, just give kind of some background information on that, and then get into heart failure, both types, systolic and diastolic, and then maybe talk about some of the imaging modalities that we can use to diagnose and further treat those conditions. So to get started, I think it's important to realize that the heart isn't quite how we always picture it, right? So we don't just have a heart that's magically beating, and I realize red would have been a better color. Rather, it's a complex system and an interaction between muscular tissue, between, innervation of your nervous system, and between an electrical system, right? So when you think about your heart beating and contracting, what's actually happening is an electrical signal. And our fancy word for this when we're talking about the heart getting signal and actually functioning and pumping that lub dub of blood, we call it depolarization. So when our heart is depolarizing, we are getting a signal. So depolarization equals signal. And what's important to talk about before we get too into that is actually the structural anatomy of the heart. So in the heart, we have two different types of main cells. We have our pacemaker cells, which make up the SA and the AV node, and we have non-pacemaker cells, which are our cardiomyocytes. So pacemakers versus non-pacemakers. So our pacemakers are what they sound like. It's our SA and our AV node. So our SA and our AV node are going to be responsible for setting and controlling the pace of our heart. So they're kind of the boss when it comes to heart rate and depolarizations, which is just the transmission of that electrical signal across the heart. They have something called inherent autorhythmicity, so they have their own rate of depolarizing, and our main pacemaker is our SA node here, and it has an inherent autorhythmicity of about 100 beats per minute. So if you just let it be, we do not innervate the heart with any sympathetic or parasympathetic activity, our heart would beat at about 100 beats per minute, whereas our non-pacemakers are just all of these other cells, right? So we call them cardiomyocyte, it's just all this tissue. And in the heart, we have something called gap junctions. So there are different ways cells of a tissue can be held together. And in gap junctions, we kind of have some space in between. So we can allow for transmission of ions, which is particularly important, and which I'll get into a second, for depolarization of the heart, fluid, but in the main thing here with the heart is that as we depolarize in that electrical signal, we use ions to do so, and so we're able to transmit that signal from cell to cell so that the whole heart can beat in a coordinated way. Because just thinking intuitively, do we want our heart, this guy is contracting like this guy is contracting like this? Intuitively, we would not survive well or function well if that was how our heart was functioning in this super sporadic manner. So that's not what we want. Now, I just talked about something called an ion, and all an ion is a charged particle. And our whole kind of electrical system, if you will, is based on these ions. So inside the heart in particular, we have three main players. We have potassium, we have sodium, and we have calcium. So naturally, we have a higher concentration of potassium inside of the cell than out. For sodium, we have a higher concentration outside of the cell than in, and same deal for calcium. And so what happens is, things like to go from high to low concentrations. Our ions are naturally, if you will, generous. They want to spread the love. And so they're going to go where there's less of them. So because, again, I just explained this, but if we have more in here, it's going to go out to where there's less of. But the problem is that we have kind of these gates where these ions can't always do what they want to do. So what controls our electrical signal in our heart is whether or not these gates are open or closed. So if we remove this gate, I didn't mean to remove all of that. If we remove this gate, then calcium can flow in. And this is changing whether this is negative or positive, right? Because these ions have charge. They happen to all be positive. And because we are keeping more of our positive ions outside of the cell, we are more relatively negatively charged on the inside of the cell of our heart. And so how does that actually play out? So again, because we have two different types of cells, our pacemakers and our non-pacemakers, we have two different types of depolarizations. So let's start off with our non-pacemakers. And I'm about to draw something called a depolarization curve. This is not perfectly to scale. This is just to explain the concept. So don't come at me for my artistic abilities, but this is kind of what the depolarization looks like. And this is our signal, and this is over time. And I actually could draw this a little bit better, so I'm going to try again. And so at different points in our graph, we have different things happening. So I kind of want to walk through what ions are doing what to cause depolarization. So at point 4, on both sides, we have our sodium-potassium pump. And what our sodium-potassium pump is maintains kind of a resting membrane potential. And all that is just maintaining that this is our normal electrical state, this is our normal charge, which is what we talked about here, where when we think of normal, we want to think of these guys as being closed, not moving from place to place, kind of in a static position. And so what our sodium-potassium pump does, well, not so much in a closed position as in a fixed ratio. And our sodium-potassium pump determines that, so we pump 3 sodium out and 2 potassium in. Because we are pumping more sodium out, which again is positively charged, then we're bringing in another positive ion, we establish that negative resting membrane potential. And when our cell's at a negative resting membrane potential, nothing's happening because that's normal. So how do we say open the door to get the ions to come in? Well, we do so by something called our fast sodium channels. So if our heart wants to depolarize and beat, which it of course does because we like to survive, we open up our channels to allow sodium come in. So we remove this guy here, not that guy. Don't remove your heart, guys. You know what? You're removing this signal right here. You get my point. And so we get what's called an influx, a rapid influx of sodium. Because sodium is knocking at the door. It's begging, let me in, let me in, let me in. So when it does that, because it's a positive ion, our charge goes from a negative end to a positive. It's called depolarization. And that's what I'm referring to if you hear me saying depolarization throughout this video. It's a very literal name. You're going from a negative to a positive pole. And this is what's actually allowing the signal for our heart to contract. So think about even turning on a light switch. You have to have that signal travel through the wires before your light turns on. This is our, logistically speaking, traveling through the wires. So when you get to the top here, our potassium channels open. And so we get an efflux because remember we have more potassium inside the cell than outside, so that potassium is going to rush outside of the cell, which is going to bring us back down because we're losing a positive ion. But instead of just immediately going back down, we actually at this point open a calcium channel, which slows down this progression. I'm going to get to it in just a second, but calcium is actually very important in our depolarization, in our generating force, that actual process. And so this slows it down because we have a positive ion entering the cell, a calcium influx, but eventually this levels off and our potassium continues to exit until we go back down because we're allowing more and more of a positive thing to leave, thus making us more and more negative. So this is the depolarization of a non-pacemaker. Contrasting that with a pacemaker cell, which looks a little different because, again, we have that inherent autorhythmicity. We don't really need that that resting membrane potential because it's kind of just been established, so we don't need as much help with that. And so their depolarization actually looks more like this. And at this point, we do have some maintenance of ions just to maintain kind of a pseudo resting membrane potential. This is actually a calcium influx. Then this is a potassium efflux. This is kind of what I just explained, but rather than having that plateau here, over here, from our calcium entering, it's just fast calcium enters the cell, boom, we go to a positive end, and then potassium exits. And again, this, particularly with the SA node, is going to depolarize, at about 100 beats per minute. This is called spontaneous depolarization. So it doesn't need, essentially all that means is that it does not need an input to depolarize. It is inherent, it is automatic nature to these guys. So we've talked about kind of what's happening on a cellular level, but let's zoom out even a little bit. So this is the heart. And so we've established that we have this electricity running through the heart. That's cool, that's great. Let's have some more information on that. So I've mentioned these two guys, the SA and the AV node. These are our two pacemakers. They kind of determine the rate of the heart, but your SA node has dominant control. I'm going to write this out again so you can see it. And so when we get that depolarization, that signal from our brain to tell us, okay, it's time to beat, which, well, we don't even get the signal from the brain because, again, this is inherent autorhythmicity. What happens is a depolarization signal that follows this pathway. So right now you're looking, this is the right side of the heart, left side of the heart, right atria, right ventricle, left atria, left ventricle. So our depolarization is going to come in, and it's going to travel across the atria. That depolarization is then going to hit the AV node. The AV node takes a little bit to get this signal all the way down. So we have something we call nodal delay. And this gives our ventricles time to fully fill with blood before we begin going out to systemic circulation. But then we hit something right here called the bundle of His. It's just a bunch, if you will, of electrically excited tissue. Our signal then goes down these guys called bundle branches. So we have left and right. So these are our right bundle branches. These are our left bundle branches. They hit the apex of the heart. Then they travel up the Purkinje fibers. And this is super important that our depolarization and our electrical signals happen in this order because that's the way our blood flows through the heart, right? So we're going, blood's entering through the superior and inferior vena cava. It wants to go through the atria. So by contracting from top to bottom, we're pushing blood down into the ventricles, which is what we want. And then by pushing back up, we're pushing blood up and out through the aorta, through our pulmonary system, so we can oxygenate our blood. So our depolarization is not random by any means, and nothing in the heart should be. It is very coordinated for a very specific reason. And one thing we can actually use to kind of look at this depolarization is something called an EKG. And I won't get too much into that, but it's just worth mentioning. It's called an electrocardiogram, depending on if you're using a American or German spelling. It could be EKG or ECG. And it just allows you to trace the depolarization. So this is our P wave, our QRS complex, and this is a T wave. This represents depolarization of the atria here. This represents depolarization of the ventricles, and then as they repolarize, which is where we have our sodium-potassium pump, corresponding to kind of this reset up here of the system, we see something called a T wave. So the faster your heart's beating, the closer this whole cycle will be to another one. And then finally, the last part is how do we actually exert the force? So we've seen that we have electricity in the heart. We have different types of cells in the heart. We have a coordinated electrical system. But how does that play out? And we can do so through something called the goldfish molecule. Supposedly, it is supposed to resemble a goldfish. So in your heart cells, which remember, guys, your heart cells are muscular. So just as your leg muscles contract, your heart muscle is actually physically contracting. So calcium enters into something called the T-tubules, which kind of just act as holding slots for your calcium. These are your L-type receptors. They have affinity, meaning they like to bond to calcium. And so what happens is this calcium binds, and then it activates your ryanodine receptors, which lay on your sarcoplasmic reticulum inside of the cell. And one of the things your sarcoplasmic reticulum is responsible for is excreting calcium. And if you'll remember, calcium is really important in both our cardiomyocytes and our non-pacemakers. Excuse my dog. So it secretes calcium from the sarcoplasmic reticulum. And so once we spit this calcium out, we have our excitation contraction coupling, our actual muscle contraction. So primarily we have our strand of myosin, and then we have our actin strand. So here's our actin, here's our myosin. On our myosin, we have 3 different types of troponin. So this is kind of similar to a typical muscular contraction of, say, your skeletal muscles, but a little different. We have 3 types of troponin. Troponin T, which is going to bind your tropomyosin to your troponin, which is what we're looking at right here. Troponin C, which binds to calcium. And then troponin I, it acts kind of as an inhibitory complex. If it is on, you're not going to get any contraction because it's blocking all of these guys from contracting. But what happens in a fully functional heart is this calcium comes in and binds to troponin C. When it binds to troponin C is released, and then eventually you release all your forms of troponin so that your actin is exposed to your myosin. So your actin grabs on to your myosin heads, and then we get the typical muscle contraction, the half a pull releasing an inorganic phosphate, you have your ADP, then you pull again, getting a full power stroke of the muscle. And that, again, same deal as if you're running or lifting weights and trying to contract, as we are just existing. Although it is autonomic and not intentional, like when we're doing skeletal muscle contraction, we still need muscle contraction to function. So this is what's actually exerting the force, and again, these are the ions that are causing these depolarizations. So all of this is incredibly interrelated.